I would like to invite our first speaker today, Professor Christian Grimm. So we have a long-standing and very fruitful collaboration with Christian, and it never ceases to amaze me how many positions Christian manages to have, being Associate Professor of Molecular Pharmacology at LMU, PI and Head of Pharmacology at the Fraunhofer Institute for Translational Medicine and Pharmacology, and he started a third position recently as a Professor of Organelle Biology and Pharmacology at the University of Oxford. So I hope that's going on in my former homeland. So Christian's going to talk to us today about high-throughput organelle electrophysiology. So thanks, Christian, please.
Thank you very much for the invitation, and it's nice to be back here to this fantastic yearly meeting. And I had already the chance last year to give a talk, and it's my pleasure now today to talk a bit more about the technical aspect of our work. And this is related actually to lysosomes. And we have already heard from Niels briefly about the increasing importance of lysosomal channels. Lysosomes are probably not, to all of you, known as signaling hubs, or that they play a huge role in transport and release of ions. Most of you will think lysosomes are degradative organelles, and that's true. I mean, of course, they play an important role in degradation of macromolecules. But in recent years, quite an astonishing number of channels and transporters has been discovered in the lysosome. And among these are, for example, TRPML1, which Niels has briefly touched on. There's a number of clinical and preclinical works going on by different companies now on this target, TRPML1, primarily for neurodegenerative disease treatment. The two-port channels are another upcoming group of lysosomal cation channels that we are very much interested in. But besides that, there's also other channels that we are expanding into a bit. TMM175 is probably familiar to at least people in the lysosome field as a channel that is with strong evidence that it plays a role in Parkinson's disease. There's a lyso-BK channel we've also published on, and TMM63 is probably the new kid on the block. It's a channel family, actually. It's three variants of TMM63, which are involved in mechanosensation. And this seems to be important for all these, when you think about lysosomes and endosomes, how they communicate with each other and how they sort of cleave off parts of it. So by fission and fusion, there's a lot of tubulation going on, which of course causes mechanical stress. And we and others believe that TMM63 proteins play a role here. TMM165, not sure if many of you are familiar with that protein. It's more transporter. And we believe that this is the protein that is importing calcium into lysosomes, something that has been discussed throughout the literature a lot, what the identity of this protein might be. Now, I don't think I have to convince you that ion channels are important in general. If you look at these numbers, you see that about 14% of all drugs, if you combine transporters and ion channels, are of all drugs on the market actually have ion channels and transporters as targets. When you look at the nervous system, this number increases even up to 49%. So a huge number of channels is really of tremendous importance for pharmacology and for treatment of diseases. And there are many programs going on, more than 500 programs worldwide on ion channels and transporters for all kinds of diseases, and more than 350 companies involved in the research in this field. So coming back to the lysosomal channels, I mean, as I said, our primary focus over the years was two-port channels and TRPML channels, and they are seemingly mainly playing a role in neurodegenerative diseases. So treatment of lysosomal storage disorders, I think this is one of the primary focuses of attention, but also more widespread neurodegenerative diseases like Parkinson's and Alzheimer's. But there's a whole range of other diseases. I don't want to go too much into detail now, but where these channels play a role. And coming back to this cartoon once again, here you also see that many of these, I already mentioned TMM175, Parkinson's, ATP13A2 It's probably a polyamine transporter, but there's also different opinions on that, what the real function is, but also clearly involved in Parkinson's disease, Batten's disease, also here, links to the lysosome, CLN7, a chloride channel that seems to be involved in Batten's disease, another neurodegenerative disease, which develops early in childhood. And you see up here also other diseases, CLN3 is also Batten's disease, and MPC1 Niemann-Pick disease. All of these are transporters in, or proteins that are associated with transport of ions in the lysosome. So that's all nice. If we want to discover new drugs, then we have a fantastic technology when it comes to ion channels. That's electrophysiology, as you all know. But is it applicable to lysosomes? Because lysosomes are so small, right? They are between 100 to maximum 100 nanometers to maximum 1 micrometer. So several years ago, and I'm very pleased to see Dr. Yukei Chao here. He was actually one of the pioneers in my lab to develop the lysosomal patch clamp technique. I just show you a video if you haven't seen this in any other talks that I did before, but to show you or give you an impression how we do this lysosomal patch clamp manually. Because you can imagine later on we will come to high throughput lysosomal patch clamp. That's something that we really want to go to. Because this manual isolation of lysosomes is very time consuming, and the failure rate is also quite high. So the first step is here with an isolation pipette to isolate this lysosome out of the cell. So before that we have enlarged the lysosome with different possibilities how to enlarge them. Apilimod, for example, or vacuoline can be used. And then we have a diameter of about, let's say, several micrometers, and that's something we can patch. That's something we can patch. But before that, we have to isolate it. We have to isolate it from the cell, and you see here that we're almost there with isolating it. Once we've done that, we keep it still attached to the cytoskeleton of the cell, because if we were to isolate it completely from the cell, it would just float away. Right? So we have to keep it attached to the cell. And then we can come in now with the patch pipette, we find our lysosome again, and then we can do like a whole lysosomal patch clamp. So, but as I said, this is time consuming. Success rate, a good day. Yeah, what was your best success rate? 2 per day? 3, yeah. But that's already very good. But that's obviously not good for, let's say, drug discovery, for compound screening, all of that. So we were funded by the German Ministry of Economy for an explorative project together with Nanion to see if, I mean, there's of course the work that Aurea does, the microfluidics approach, right, to isolate lysosomes. But we also thought maybe there's another method we could use, and that's simply ultracentrifugation. And here's sort of the protocol we came up with, how we step by step through differential ultracentrifugation, we end up with a preparation that mainly contains lysosomes, which then can be used, for example, on the SyncroPatch to be patched there and to be used in a high throughput scenario. Now, here, please focus on the orange bar. This is really the, let's say, quality control of our preparation. We have different markers for different kinds of organelles, including mitochondria, for example. Don't want to have mitochondria in there, right? All kinds of other vesicles and ER, parts of ER, plus a membrane. And for that, we have markers, right? We have, for example, markers for ER, for Golgi, for nucleus, for mitochondria. And these up here, LAMP1, LAMP2, RAP7, cathepsin D, these are markers for lysosomes. And if you focus on the orange fraction, and that's actually the last fraction before we have fairly pure preparation at the end, you see that we have really enriched here all these markers for lysosomes quite nicely. We do have barely any Golgi left. We have barely mitochondria, nuclei are gone. Of course, they are large and heavy. Also, plasma membrane is, we've gotten rid of plasma membrane mostly. We still have a little bit of, sorry, that was one too fast. We still have a little bit of the plasma membrane here. Sorry, pancreatin is a bit less. Yeah, and here the sodium, potassium, ATPase is still a bit more. So we have still some impurities, but we have really gone a far way here from the beginning here, where we had a lot of impurities still in there, to the end where we have an almost pure lysosomal preparation. And this compares also quite nicely to the microfluidics approach. If you look at here with the red arrow, this is basically how they were able to reduce things like PMCA, it's a marker for plasma membrane, calnexin for ER, mitochondria. So it's comparable here that you get rid of these components and you still have most of your, or you have enriched your lysosomal partition. So the last step we figured out, which is really important at the end of this process of ultracentrifugation, is that we filter our samples, because that really gives us at the end these nice isolated lysosomes, which are enlarged roughly to 3 to 4 micrometers in the best case scenario. There's also smaller ones, a few larger ones. And of course, we quantified that, and you can see here, so the largest fraction is between 2 to 2.5 micrometers. That's still patchable. Of course, we would like to have it a bit larger, but you see here 17.9%, 12.5%. These are really the ones that we love most. They're fairly large. They're in part larger than 3 micrometers. And that also compares fairly nicely to what is achieved with the microfluidics preparation, where also there are some really large ones, a bit more than we have. 6 micrometers we have rarely. But you can compare these fractions combined are roughly corresponding to what we have here in these two last fractions. Okay, then how does this look like in action then when we use the SyncroPatch? Can we really measure reliably currents with the SyncroPatch from these isolated lysosomes? The answer is yes, we can do that. And we have a success rate at the moment. We still hope to improve this further at the moment, between 40 and 50%, which is not too bad. But we're hoping to get higher here, but then we get really nice currents. Here you see, for example, this is an example for TBC2 with PIP2 activated. We get a current that looks like TBC2. It's blocked with ATP. That's something also which is a feature of TBC2. So maybe it is TBC2. Yeah. So it's like a dog, right? It looks like a dog. It walks like a dog. Maybe it is a dog. And so we were quite happy to see that. And when we compare that directly to manual patch clamp, how do the currents look like in the manual patch? You can see here that this is with the Nanion solutions. So they differ a bit from the typical solutions that we use. So we use, for example, much less calcium normally in our patch solutions. Nanion uses higher calcium that is needed for the gigaseal formation in their settings with their chip technology. But you see, when you compare these currents, so this is with the same solutions in the upper row, right? This coming from the SyncroPatch and this coming from manual patch clamp. And so the reversal potential seems about the same, right? And we can actually get currents, obviously, in the SyncroPatch, which look very similar. This is with our regular solution. See, there's a bit shift in voltage in the reversal potential, but all in all, it looks all very comparable. One other thing that's interesting here on this slide, just to mention it also when it comes to maybe TBC2 as a drug target, because TBC2 is very stable when it comes to pH. There's ion channels which are very sensitive to pH changes. TBC2 seems not. Its activity barely changes when you look at 4.5 versus 7.2. And that is very different when we look at TRPML1. And as I said it earlier, TRPML1 is one of the channels that's probably mostly explored at the moment by pharmaceutical industry. But one critical point actually regarding TRPML1 is its sensitivity to pH. You see, when we go to pH 7 here, we have much lower current density as compared to the situation at 4.5. We have also tested other drugs in this system here, again, going back to TBC2, now in direct comparison, urea samples versus the UC or ultracentrifugation samples, this time stimulated with a small molecule called TBC2A1P, so agonist 1, P-type, PIP2-like type, because there's two different modes of activation, NADP versus PIP, and this is a PIP-type compound. And you see that this also compares nicely between the urea prep and the UC prep. So we can activate with A1P, we can block with ATP, and that is pretty much the same in the urea prep on the right side. And also when it comes to the success rate, this is also comparable. So we have the success rate of the lyso catch, as we call it, then going whole lysosome and the actual current detection. And you see here that also these success rates are fairly well comparable between the two, the microfluidics and the ultracentrifugation prep. Okay, coming to the conclusions and what are the next steps? I said the success rates are at the moment in the range of 40 to 50%. We want to increase that, and we're working on that. At the moment, the currents look similar to currents obtained with manual LysoPatch clamp. That was very important to us to see that, both in size and shape. And the next steps are we want to start with a high throughput screening, actually with one of the 2 port channels, because there's lack in chemical matter that we want, this gap we want to fill. And TPC1 is a channel that is actually hard to bring to the plasma membrane. This is sort of a trick that you can use, of course, for drug discovery. You just bring the channel to the plasma membrane, but then you always need to keep in mind that the protein composition, the lipid composition is so different in the plasma membrane as compared to the lysosome. So you could pick up something that is actually not working in your lysosomal scenario later on. That's why we would really prefer to do this in the lysosome context, and TPC2 we thought is a good target for such a ATS or high throughput drug screening. We also can, with these preps, obviously can do proteomics analysis. This can be also very interesting in a disease context. So independent of patch clamp, actually, we would isolate lysosomes and then compare how the proteome has changed, for example, in a Parkinson disease model compared to a healthy model, and then see if we can reverse this phenotype maybe when we apply certain Parkinson treatments, drugs. And that's also something we can do with these organelles. And and that's basically the summary in one slide of the applications that we can really, or the forms of applications of these isolated lysosomes. Automated patch clamp is one thing. We can also use it with the SSM. We have done some pilot experiments here already together with Nanion. And of course, all kinds of functional assays from enzyme activity measurements to lipidomics, proteomics. And I think this is really important for the field and especially in the disease context. And with that, I hope I have convinced you that lysosomes are important. We are happy that we can really contribute to moving the field forward. Also in terms of seeing hopefully more drugs coming, or lysosomal ion channel drugs coming to the market at some point. I would like to thank my laboratory in Munich, especially David Yashlan, who's also here in the audience, and Nitish Sadanandan, who has done most of the work that I've shown you today, and Niels, of course, and Marcus for their long-term collaboration, some of our papers, and thank you so much for your attention. I'm happy to take your questions.
Thanks very much, Christian. That was wonderful. Any questions from the audience or any questions online? You can write them in the chat. Any questions from the audience?
Thanks, great talk. Not so much a science question, but talking about scalability, market adoption of organelles on high throughput platforms, time and cost. So time to set it up, and then typical cost point per well, test point per well. What is that? Did you have anything you can share on that? Cost,
well, I'm sure Nanion will be more than happy to answer in detail this question, but what we figure out is that when we do a screen of, let's say, 30,000 compounds, a plate costs roughly, please correct me, Niels, if I'm wrong, 300 to 500 euros, right? One plate, 300 is more. Maybe there's also a discount when you buy more. I don't know. Anyway, so yeah, then you can calculate. I mean, if you have a 50% success rate, let's say 384 well plate, you want to test 50,000 compounds, you can calculate how much roughly this would cost you.
That would be one concentration per well, or?
That would be only one concentration per well, yeah.
Very nice talk and cool technologies.
I was wondering if you could comment on, like, you know, any technical bias that you might introduce by enlarging the cells and just picking up the largest ones.
Yeah, this is a common question, of course. It's always sort of yeah, you have to balance the problems, right? I mean, is it better to, if you, let's say you're afraid of enlarging it would sort of lead to a fusion of lysosomes with endosomes. I can say no to that. We have never seen that. So it seems it's really homotypic fusions. So lysosomes fuse with lysosomes, early endosomes fuse with early endosomes. How do we know that? We know that because we have different markers, right? RAB5, for example, is more early endosomes. RAB7, LAMP1 are more lysosomes. We never see a mosaic structure in our enlarged organelles or in enlarged, let's call them endolysosomes, right? They seem to be fairly homotypic, right? So lysosomes remain lysosomes. There's nothing mixed in. And that gives us, let's say, at least a good point in saying we still have lysosomes. They're just larger. Yeah? And of course, you can talk about mechanical stress or things like that, or osmolarity or things that could change. But I think we are less worried about that when it comes, for example, to a high throughput screen to discover new drug or new compounds for a particular channel, as compared to when we do this in the plasma membrane. Because this, as I said, the situation in plasma membrane is so different. There could be proteins that you miss really, which are in the lysosome, which are, for example, blocking the channel or enhancing the channel activity. RAB7 is, for example, a strong enhancer of TBC2. It doesn't occur in the plasma membrane. So this gives you already completely different dose response curves then. And EC50s and all of that. So this can be very misleading then when you do this in the plasma membrane. That's why I think even having this in mind, you're enlarging them, yes, but I think this is still the better problem to deal with than having simply a lack of a better alternative, I would say.
Hi, Christian. Very nice talk. Do you have already proteomics data from your lysosome, and are you also able with your technique to isolate lysosome from iPS-derived dopa neurons, for instance?
Exactly, that's where we want to go to, having different Parkinson disease models and having proteins compared in these models with a wild-type isogenic control. We have a first run. We had a first run where we did find the typical lysosomal proteins like LAMP1 and all of that, and we can compare that to the wild types, obviously, and then see is there anything up or down regulate. So we are capable of doing this with this isolation method. Of course, doing it with ultracentrifugation or with microfluidics allows us to have many more lysosomes than we would have with just manually isolating. So our first attempt was, because we thought this is the absolute gold standard, to just isolate them manually with the isolation pipette, just what you've seen on the video, right? It's a bit time consuming, but isolation you can isolate roughly, maybe Nitish, you correct me, 30 or so per day, right? 30 lysosomes per day. And so in like 10, 20 days, you're ending up with 600-700 lysosomes, and that you can use for proteomics.
Thank you.
And this we compare, of course, then that's the plan, with the UC and with the urea samples to see if we get similar proteins.
I think we have one online.
We do. To do with contamination in the lysosomal preparation, did you ever try to express the lysosomal and plasma membrane ion channels in the cells to show that you recover only the activity of lysosomal channel in the recordings?
I don't think right now that we have done. I don't think we have done that. But it's certainly, thanks for the idea. Certainly something we can try. No, we have not done that.
Okay, if there's no more questions, I think we will move on. Thank you very much, Christian.
Thank you.